The overall goal of this procedure is to image live cells through growth and division using fluorescence microscopy so that the effect of cell history and ancestry on cellular development of bacteria can be studied. This is accomplished by first transferring the cells from liquid medium with relatively high nutrient concentrations to liquid starvation medium. Next, a microscopic slide on which the bacterial cells will grow into a microcolony monolayer is prepared. Then, a time-lapse fluorescence microscopy movie is recorded. Finally, the movie is processed and the data is analyzed. Ultimately, results can be obtained that show development of a single bacterial cell through time-lapse fluorescence microscopy.
The main advantage of this technique over existing techniques such as flow cytometry and standard microscopy is that it allows the monitoring of protein dynamics, cellular behavior and development of single cells in time. Demonstrating the procedure will be Imke de Jong, a PhD student from my lab.
To prepare B. subtilis, inoculate cells from -80°C stocks in 10 ml of time-lapse microscopy, or TLM, medium in sterile shake flasks, supplemented with antibiotics if necessary. Grow the cells overnight at 30°C and 225 rpm in a sterile shake flask. The following morning, dilute the cells 1 to 10 in pre-warmed chemically defined medium, or CDM, without antibiotics. Grow the B. subtilis cells to mid-exponential phase, which takes approximately 4 hours. Measure the absorbance of the culture at 600 nanometers and dilute the cells to an approximate A600 of 0.035 using CDM.